Science Challenge of the Month talk, although we are in June, because last week was half term, so we decided to shift it to today. So Bridget actually came a few years ago as a postdoc, joined us on this campus, but in 2023 got BBSRC Discover Fellowship, that is in Center for Ecology and Conservation, but based in the SI. Bridget's work is around how bacteria evolve and interact with the environment, focusing on soil bacteria that associate with plant roots and promote plant growth. And this is really with the aim of understanding plant microbe interactions so that we can use microbe as a tool for sustainable agriculture. So today, the title of the talk is Growing Plants Sustainably, Harnessing the Power of the Soil Microbiome.
Great, thanks, Kimmy. Yeah, thanks for this opportunity to, I guess, think about my work in a different kind of framework with the challenge of the month. And I'm really excited to talk to you all about my research and hopefully convince you that microbes and especially bacteria are really cool, but also really important. So I need to learn more about them. Great, so in around 2015, the United Nations put together around 17 different goals, sustainable development goals, and these were all about kind of improving the prosperity and peace of people across the whole world. And the goal that's particularly relevant for my research is goal number 2, which is all about zero hunger. So in a longer term, it's all about ending hunger, achieving food security, and improving nutrition while also promoting sustainable agriculture. And so I'm sure you're all quite aware that feeding the world is a huge challenge, and importantly, hunger is a huge problem. And so it's kind of estimated that food insecurity affects about 1 in 3 people, and predictions that by 2030, over 600 million people will face hunger. So it's a real huge problem, and as well as calorific intake, there's the challenge of malnutrition. So this affects women a lot of the time with anemia and iron deficiency, but also children. So the first 5 years of development are really important, and lack of nutrients, but also food can lead to lifelong kind of problems with stunting and wasting. But also at the other end of the spectrum, there's a huge challenge with obesity. So in general, the whole food system is a big challenge, but there's a lot of things that we need to improve. And also a major barrier is also the price of food. And so for a lot of people, it's really hard to access affordable food. And the recent kind of global events, including the pandemic, but also some wars, have really made this problem a lot worse for some people. And in 2021, like 1 in 5 countries had trouble kind of acquiring affordable food. So it's a huge big issue. And so to kind of try to address this goal, the UN have these smaller targets. And so there's like almost 150 of these targets across the 17 goals. And the 5 that are relevant to the ending global hunger include universal access to safe and nutritious food. So I guess getting food to people. And I guess to approach this target, I guess is about production of food, but also it gets into a lot of kind of bigger issues about supply chains, politics, economics. These are really big challenges, and it also kind of relates to the fact that a lot of food, once it's produced, is lost from the food chain, from pests eating it, but also just general losses. So lots of areas that need to kind of be addressed. And the second target is all about ending malnutrition, making sure that when people have food, that this food is nutritious. The third target was all about increasing the productivity of small-scale farmers. And so a lot of food insecurity affects people in rural areas and maybe low-income countries. So it's an important aspect is empowering small-scale productive farmers so that they can provide for the communities and really help in areas where access to food is a problem. And then target 4 is that's what I'm interested in addressing. And this is all about how we produce food and making sure that these systems are resilient, but also sustainable so that they can continue for many generations without negatively impacting the environment. And then target 5 is an exciting area for people interested in genetics and biotechnology, and also the ecology and conservation department, because this is all about making sure that we maintain and also make use of the genetic diversity that exists for food production. And this is important for making sure that crops are resistant to parasites and pathogens, and ensuring again the sustainability of food production. And so, as I mentioned, my research hopes to build knowledge to help make sure that food production is sustainable. So, in regards to producing food, some massive increases in crop production happened during the 20th century, and this is what's commonly kind of referred to as the Green Revolution. So essentially, a few things happened that meant that we had the technology and infrastructure to massively increase crop yields. And so the things that influenced this included the development of particular varieties of crops, mostly rice and wheat, and a bit of maize, corn as well, that were high yield. So these were often hybrids, which kind of combines the best of both worlds for different types of plants, and they were just inherently more productive. They also, the widespread use of chemical fertilizers became a possibility, which meant that we're able to add these fertilizers to crops and to kind of help them to grow better. And there was also a lot of infrastructure around providing water to these crops, so irrigation became a big thing. And mechanization was really important. So this is like the use of on-farm machinery like tractors, harvesters, things that kind of made farming able to be scaled up more. And also the introduction of pesticides, which kind of dealt with the things that like to eat these crops. Essentially, these different things were quite common in countries like the UK, and we saw these increases in production happen across the 20th century. But there was this American agronomist called Norman Borlaug, and he really helped to make sure that all of these advances in technology, science, I guess, as well, were spread to all parts of the world. Essentially, Norman worked in Mexico, and he actually developed some of the high yield varieties by creating hybrids of wheat plants. And then he was part of the team that was funded by a lot of American philanthropist organizations that brought all of these technologies to lower income countries like Mexico and India. And for his efforts, he won the Nobel Peace Prize in 1970. But essentially, all of these technologies meant that millions of people were saved from hunger. It was an amazing story of, making a lot of small economies viable, increasing the GDP. There was a lot of benefits for these technologies. And I'm sure you can hear the buck coming. But yeah, this was a huge, huge progress, really important, making sure lots of people had food to eat. And yeah, this guy Norman was a big part of that. Here's the buck. But of course, we know that these practices are not sustainable. So essentially, we've kind of broken the negative impacts into two components. Essentially, the kind of widespread adoption of using chemical fertilizers and also growing maybe the same type of crop as a monoculture. They're really damaging for biodiversity because it kind of removes, farmers weren't growing like the less abundant, more local varieties. As I mentioned, developing irrigation systems was a really important part of making sure these crops could grow, which negatively impacted the water systems in a lot of communities. Water scarcity is a huge side effect of this massive agricultural push in these areas. And of course, when we're adding these chemicals to the environment, They're often added in excess, and then they can run off into local waterways or other systems where they essentially are toxic and they disrupt the normal functioning of these environments. So there's a lot of damage for the wider environment from this kind of green revolution. And it's also been really damaging for the soil. So essentially, what the green revolution enabled to happen is that people could constantly grow crops and constantly extract from the environment. So by adding nitrogen, this was usually the limiting factor for plant growth, but by adding it to the environment, farmers could crop frequently and constantly extract and deplete the soils. And as a result, all of the important components that are present in smaller amounts, like the trace elements, these all became depleted from the environment. So essentially, a lot of these soils that kind of benefited initially from the green revolution, they're now degraded and they are depleted of nutrients. So essentially, initially it was an amazing approach that meant that a lot of land could be saved from deforestation by increasing the yields on farmable land. But as a result, now the soils are degraded, so they've had to find new areas to farm because of these exploitative practices. Like the goals for agriculture remain the same. We want to increase the yields so that we can feed a growing population. But it is really important, from the lessons we learned from the Green Revolution, that we reduce the chemical inputs because we know that these can have negative consequences. And we also need to work really hard to make sure the soil is fertile and it maintains a healthy kind of balance. And so obviously the solution or approaches to address this challenge are really complicated. There's lots of things like policy, infrastructure, economics, all of these things weigh into it. But a major focus that everyone seems to agree on is that we need to focus on the soil. The soil is really important, and especially soil health and quality. So what is soil? Well, soil is pretty much comprised of 5 different ingredients. So we have minerals, and these are also like the particles, and these, you might have heard about sandy or silty or clay soil. These give the soil its texture and its kind of overall properties. There's organic matter, which is really important. So this is carbon and how soils can store carbon. And this carbon comes from the breakdown of plants and animals that live in the soil. There are a lot of gases that come out from the microbes, which I'll get to soon, and the functions that they carry out. Importantly, soil carries a lot of water, which is important for the organisms, plus also the wider environment. And the most exciting thing, in my opinion, about soil is that it's full of living organisms. So this includes small animals like insects, nematodes, spiders, lots of things like that, but importantly, it carries microorganisms. Right, so the majority of the organisms in soil are microorganisms. So soil is one of the most complex and biodiverse environments for microbes on the planet. And it's really important, and the microbes are really abundant. And 1 single gram of soil has about 100 million, up to 100 million different microbes. It's a hugely important, diverse ecosystem. And the composition of soil, if we look at the breakdown of the microorganisms, it's mostly bacteria, but there's also fungi, viruses, and other single cell eukaryotes like protozoa and algae. And these microbes drive soil function. So the microbes essentially are the soil because they capture and cycle the nutrients for the more broad ecosystem. So essentially, soil without microbes, I'd argue, isn't really soil. So yeah, as you can tell, I love microbes, and I want to emphasize that healthy soil is full of microbes, and if the microbes are happy, the soil is happy. So I guess there's this idea of what is a healthy soil. Well, I guess because of the complexity of soil and lack of understanding about what the microbes are doing, this idea is kind of quite hard to define. But in general, like a lot of biodiversity and kind of ideas, a more diverse soil is considered a healthy soil. This is a big area of research to try to figure out what do we want our soils to look like for them to be healthy. And so then I guess bring in the green revolution and the agricultural practices that are still quite commonly used. How do these practices affect the soil microbiome? Well, ultimately they tend to reduce microbial diversity and also change the composition of the microbes. And there's a few ways that they do this. So firstly, the addition of chemical fertilisers alter the pH of the soil, and most microbes will have a window that they function at most effectively, and by increasing the pH, some microbes won't be able to carry out their normal functions and exist. And also the addition of nitrogen to the soil kind of bypasses the microbes, and it replaces a normal function that they carry out of capturing and cycling nitrogen normally. It replaces this function, and so we find that microbes that can carry out this function are no longer needed, so no longer grow in the soil. The use of machinery for tillage and ploughing and cultivation, as you can see in the picture, it disrupts the soil structure. So essentially microbes that might have been growing happily under the surface of the underground surface are now exposed to a completely different environment with oxygen, so they're not going to be very happy. Also, this process of disrupting the soil structure can release carbon from the soil, and we know that a really important function of soil is to capture and store carbon. And also the addition of chemicals, particularly pesticides, are actually toxic to the microbes, so they won't be able to grow and carry out their functions normally. So essentially a lot of the practices are pretty bad for the microbes, and they're not, and ultimately they tend to kind of bypass the microbes and interrupt the normal functions and relationships that the normal functions that the microbes carry out, but also the normal interactions that they naturally have with plants. How do soil microbes interact with plants? Well, essentially, when a plant grows in soil, it kind of changes the environment for the microbes because the plants excrete a diverse array of molecules from the plant roots, and they create this nutrient-rich area surrounding the roots, and we call this area the rhizosphere. And essentially, different plants will release different types of molecules, and the composition of these molecules will determine which soil microbes will assemble to form what we call the rhizosphere microbiome. And because this environment is very nutrient rich, these microbes that live on the plant roots and the rhizosphere, they're a lot more active than other microbes in the surrounding soil. So it's a really interesting, important environment to study. And because of all these nutrients, the rhizosphere microbiome is a lot denser than the surrounding soil. So it's kind of like the place to be if you want to be a micro from the rhizosphere. So lots of different microbes will grow in the rhizosphere, and some of them are especially important for plant growth. So we have mycorrhizal fungi, which help to extend the plant roots to increase nutrient uptake. We know about rhizobia that live on the roots of legumes, forming a symbiotic relationship. But in this talk, we're going to talk about plant growth promoting rhizobacteria. So these are a diverse group of bacteria that live on the rhizosphere, and they form a mutually beneficial relationship with plants. So the plant gives the microbes lots of nutrients to grow on, and in turn, the bacteria carry out functions, diverse functions that might help the plant to grow. So these functions can range, some examples are here. So these beneficial plant growth promoting rhizobacteria may make nutrients more available for the plants so that they can grow better. They may restrict pathogen access to the plant so that it can avoid infection. or they may actually produce molecules that actively stimulates the plant to grow, again, helping the plant to grow. And so back to our future of agriculture and trying to make agriculture more sustainable, there is a lot of enthusiasm for using these beneficial plant growth promoting rhizobacteria to sustainably promote plant growth. And so in this picture, this shows examples of what happens when we can grow these bacteria and add them to crops as what we call biofertilizers. And as you can see, there's some striking evidence that adding these bacteria is really beneficial for the plants. They grow a lot better. And so there's a lot of enthusiasm for using these bacteria as a tool. However, the results can be inconsistent because there's still a lot we don't know about the biology of these bacteria. Because as I mentioned, the soil is complex and exciting, so there's still lots to learn. And so one of the reasons that I think that, when we apply these beneficial bacteria, they're not always effective at promoting plant growth, is that we are missing a key understanding of lots of about how these bacteria interact with the environment. So if we think about when we apply these beneficial bacteria, there are several criteria that need to be met for the bacteria to be able to promote plant growth. So firstly, they must be able to grow in the particular soil type that they're added to. I mentioned before that, some microbes have a narrow pH growing range, so it's not guaranteed that they'll actually be able to exist there happily. They need to grow on the particular roots that the plant root exudates that the plant excretes. Different plants have different profiles of root exudates. And then once they're actually growing in the soil or on the plant roots, they then must compete with the other microbes that are hanging out on the plant root surface. So there's a lot of things that these bacteria must do to be able to be beneficial for the plants. And so this is what my research aims to address. So I'm interested in understanding how bacteria evolve and interact with the environment, and in particular in the soil and in the root surfaces, and ultimately to determine the set of traits that the most beneficial bacteria have. And this will ultimately inform how we apply them to crops as beneficial bacteria via fertilizers, and allow us to select the most beneficial strains for use. But also fundamentally, I'm interested in gaining insights into how bacteria exist and function in the soil and the rhizosphere. So how do we study these beneficial soil bacteria? So there's kind of two main approaches, and from my perspective as an experimental microbiologist, caveats always. So the first one is to use microbiome level bioinformatics. And so this involves kind of sampling the whole soil environment and using a lot of bioinformatic tools to look at the whole community. And this can include using metagenomics to look at the genomes and the genetic material of the entire community to try to understand which organisms are present and which particular traits they might encode to get an idea of what their potential function could be. You can also use metatranscriptomics. So this involves looking at what the microbes are actually doing, which genes they're expressing to kind of understand what they might be, their functions might be. And we can also use mathematical modeling to try to understand these complex interactions and gain, genetic predictions about what sort of, the influence of different factors in this complex environment. And so the benefits of these approaches is really that we can capture the entire community. It's very complicated. A lot of microbes depend on each other to carry out their functions. So by using these large-scale tools, we can study the community in its entirety. But one of the challenges is that we are only kind of observing what's happening, and it's hard to properly attribute causality for specific events that are happening in the soil. The second approach is to use a model system. So this is when we take a specific bacteria or maybe a small group of bacteria or microbes that we study as a kind of representation of the wider community. And this means that we have a tractable system for testing hypotheses. We can manipulate the community in known ways to test the influence of a single variable. But obviously the downside is that this is just a small community and it kind of lacks the realism of the larger soil environment. So both of these two types of approaches are really useful, and kind of using both together, that's how we will kind of figure out what these soil microbes are up to and make fully informed decisions about biofertilizer use. But the main one I use is model systems. So I work with bacteria from the Azospirillum genus, and these are present in all soil types, almost all soil types. And they're from the alpha proteobacteria group, which means they're kind of part of one of the most abundant microbe group in soil. Importantly, they associate with diverse plants. So studies have shown that they live in the rhizosphere or promote the growth of plants from 25 different botanic families. And importantly, they've been used as a biofertilizer for over 50 years in South America. So they've got a strong track record of being applied successfully to promote the growth of wheat and other crops. And importantly for me as a microbial geneticist, they're genetically available. So they're straightforward to grow in the lab, but also we can genetically manipulate them to test generate. And so today I'm going to tell you about 3 ongoing research questions in my group, which is currently me, soon to be 3, which will be exciting. So we'll first look at how do bacteria evolve in the rhizosphere. So one of the great things about bacteria is that they can rapidly evolve to adapt to new environments. And they do this by acquiring mobile genetic elements. So these are discrete regions of genetic material that can move between bacteria and transfer beneficial traits between cells. And so this figure here just shows that there are a range of different types of elements, the details of which aren't super important, but they can transfer between cells via different mechanisms. And importantly, they can encode a range of traits. So for example, a mobile element may encode genes that provide virulence traits to the bacterium, so it can now live in a new host and occupy a new niche. It may encode genes that enable it to grow on a new substrate. So these can be really, life-changing acquisitions for bacteria. And importantly, these mobile elements can be really abundant in some bacteria. So in some species, over 50% of the genes are acquired on mobile elements. So they can really contribute to the overall bacterial characteristics. However, acquiring mobile elements, while it can be good, it can also be really bad for the bacteria. It can disrupt how they function. So bacteria carry defense systems that limit element uptake. And so these defense systems, they're really abundant, they're really diverse. They function through a lot of mechanisms. I won't go into all the details now. It's a whole other field of research that happens here in the ESI. But essentially, they can kind of function at different stages of the cell. So the bacteria may prevent them from getting into the cell. And once these elements are in the cell, they may cut them up, degrade them. Or if things get really bad, the bacteria may have to induce cell death to prevent these elements from spreading between cells. So quite dramatic consequences for these bacteria. And one single bacterium will usually have around 10 different systems. And importantly, the distribution of these systems does not always match with the host phylogeny, meaning that they're frequently lost and gained from the genomes, including on mobile elements. There's a lot of things happening. And so due to the recent advances in sequencing technologies and bioinformatic approaches, there's a new appreciation of the diversity of mobile elements and defense systems. We have more evidence that in all environments, bacteria are constantly exposed to mobile elements, meaning that they frequently interact with the defense systems. And as I mentioned, the outcomes of these interactions can really affect the fate of the host cell. So they may evolve, acquire a new element and occupy a new niche, or they may die. So it's really important And while we understand the particular mechanisms of these systems, the ecology and evolution of these interactions is well understudied. So this is what I've based my career on, understanding these dynamics. And so I won't go into these details more for the interest of time, already running a bit late, but essentially my PhD and postdoc really helped to enhance our understanding of the interactions between defences and mobile elements. So I showed that a particular type of defence system may inhibit some mobile elements, but it may also enhance the uptake of mobile elements, just depending on what the particular defence system targeted. So real opposing effects that it can have on the bacteria and the fate of the bacterium. And I showed that a particular type of defence system may function differently when it's in different bacteria, which really highlights that it's really important to study different model systems and in different environments to fully understand how they might function. And when I joined the ESI as a postdoc, I looked at how the environment influences mobile element defense system interaction. And so I used a host that could use one of two different defense systems against a particular mobile element. And I saw that the environmental factors influence which defense systems it used, and that which defense system it decided to use had really important consequences for the fate of the host cell. So how do I take my knowledge and my background research and then apply this to the rhizosphere? That's essentially the goal for my fellowship. And I guess we can make some predictions. So as I mentioned, the rhizosphere is a nutrient-rich environment. It's a hotspot for microbial activity. So we predict that mobile elements spread a lot in this environment, but it's also a highly competitive environment. So it's likely that, once the cell receives a new mobile element, selection will act quickly. And so there's a lot of selection happening in this complex environment. But given that mobile elements will spread quickly, the role of defense systems is unclear. So no one has studied how defense systems function in a cell in the rhizosphere. It's a huge knowledge gap in the field. And genomic studies that have looked at how defense systems may shape mobile element uptake over time have conflicting results. So there's evidence that some systems may stop mobile element transfer and ultimately bacterial evolution, whereas for other systems, there's no evidence that they influence bacterial evolution. So lots of open questions and a really exciting environment to study these in. And so, as I mentioned, I wanted a fellowship to essentially address this question, to mediate my transition from using lab model strains to this complex rhizosphere environment. And I've got just under a year left of funding. And so one of the reasons that I chose Azospirillum as a model system is that it's really relevant to this particular research question. And this is because a study by my collaborator just over a decade ago showed that Azospirillum evolved to be able to grow in soil and on the rhizosphere, diverging from its aquatic ancestors by acquiring all the traits it needs on mobile genetic elements from other soil bacteria. So it kind of did this massive influx of relevant traits for this occupying this new niche that it lives in. So as a result, it has a large genome, which is comprised of multiple components, which is quite unique in bacteria. And so given this life history with this massive uptake of mobile elements, it's not clear the role that defense systems have played in this evolution. But I predicted that they have fewer defense systems that enabled this massive genome expansion. And so the approach that I've used to answer this question involves some genomics analyses and then statistics, and then ultimately I will test these predictions using genetics to make mutants to directly test these phenotypes. And so the first step was to generate a library of Azospirillum strains. So when I first started there was about 100 genomes available in the database, but only 20 were complete. And so I wanted to generate a library of strains from UK soils that were hopefully better adapted to grow in UK conditions so we could apply them to UK soils. And so I worked with a range of soil types, as you can see here. And so I was lucky to get soils from a collaborator that were kind of collated as part of a larger project. And so what they did was they took soils and grew a range of crops in them. And so I received soil samples, rice-seed samples of wheat, so hopefully a natural environmental enrichment step, plus the equivalent bulk soil. And I've been using an isolation method using kind of semi-selective differential growth media. And to date, I've isolated well over 100 strains and sequenced just under 100 of these. And by just looking at the genomes, I can see that there's enormous genetic variation. So they range in size from 6.3 to 9.4 megabases. And as I mentioned about all the different components of their genome, this number ranges from 5 to 12. And so I first looked at how these strains relate to all the other known Azospirillum genome sequences. So I generated a phylogenetic tree of these bacteria using Kangaroo and then FastTree. And just to orientate you to this tree. So in the black, we have the different sequence global isolates. And then in the orange, we have my novel UK strains. These colours kind of group strains from the same soil sample. And then we've got two colours of these lines. So in blue, we have the bulk soil. And then in orange, we have the wheat rhizosphere. This one has a little Cornish flag because they're from Cornwall. I isolated them from my garden soil, which is really exciting. Essentially, what you can see is that samples from the same soil sample cluster, but then these UK soil samples are more related to other non-strains than to each other, which suggests that there's no clear UK signature of Azospirillum, and that my strains kind of span the diversity of all known Azospirillum, maybe extending at these areas. Perhaps I have missed some new species. So it's really exciting to be able to add to this global collection, but also find exciting new things in the UK. I've been using, to try to understand defences and mobile elements in these strains, I've been using genomics tools made by researchers across the world that have made these amazing tools that allow me to make predictions about what these strains may encode. These are all listed here. And so what I've found is that these strains carry lots of mobile elements and defense systems. Firstly, looking at defense systems. So this is kind of a figure to show the distribution of these systems. And across the top, you can see lots of different systems are represented in these strains. They have lots of different numbers of systems from 4 to 13, but again, kind of spanning the average of 10, which is reported in the literature. And they vary in the number of copies of each of these systems. But I guess the interesting thing to know is that these strains have different types of systems, but also within this group of closely related bacteria, some strains are present or absent from their close relatives. And this is quite common because these defense systems can frequently be lost and gained from the genomes. So then I looked at mobile elements. These are just 3 different types, and each of these dots represent a genome. And you can see that the numbers of these mobile elements varies a lot between the strain. That's kind of a recurring theme with this work, is there's a lot of diversity and variation. That's very exciting. There's lots of questions arising from it. And so, as I mentioned, Azospirillum kind of diverged from its ancestors to be able to promote plant growth. So I thought if I wanted to understand the influences of defences on mobile elements in Azospirillum, I should also look at its relatives. And so this is what I have depicted here. We have Azospirillum, the particular genus I work with, plus also relatives from the family level in green. There's only 34 of these. Plus as an outgroup, I have bacteria relative at the order level, bacteria from the Acetobacter family, which is quite interesting. They're acetic acid bacteria, so they kind of ferment kind of found isolated from like wine and acetic acid. Quite a different niche there. So essentially I've been analysing these 3 groups. And so what I found is that by looking at the genome size, Azospirillum have larger genomes, respectively, than their relatives at the family level, and these are much bigger than their order level relatives. And it kind of correlates also with the number of genes. When I look at mobile elements, as expected by their larger genomes, Azospirillum have more different types of mobile elements. But again, there's a lot of variation between these genomes. But then when I look at defense systems, I found that Azospirillum have fewer than expected defense systems based on their larger genome size, which may indicate that, that they kind of have fewer defenses, and this maybe mediated their evolution. There's a lot of work ongoing with this to try to do some proper statistical analyses. And yeah, but I didn't want to keep you fully in suspense. So I kind of did some very quick plotting. Here I've plotted all of these different genomes for these different groups. And I plotted the total number of mobile elements against the number of defense systems. And you can see that there's no obvious trend, which means that at this large scale of total number of elements, defense systems don't seem to have influenced mobile element acquisition. However, the elements that we are detecting in the strains in the present moment, perhaps, well, these are kind of maybe recently acquired elements, and if they've acquired them over time, maybe they're no longer recognized as elements. So I used another proxy for mobile element and evolution by looking at genome size. So this is the length. This is the length of the genomes. You can see that the order level relative have much smaller genomes. But again, there's no strong associated between number of defense systems and the genome length. So yeah, as I said, this analysis is ongoing. But to summarize, I've increased the number of completely sequenced Azospirillum strains, and I've predicted these different components to try to understand the evolution. The next steps will be to work with a really talented PhD student in the Western group called Joe, who will help me with some complex-ish analyses. And then I hope to experimentally test the predictions from these models. Okay, so the next question. So I guess my introduction to the rhizosphere was looking at the evolution of these strains. But now that I've established a model system, most of my research will focus on understanding these plant-bacteria interactions, starting with which traits are most important. So as I mentioned, there's a lot of diversity in this library of Azospirillum that I have. And so I wanted to understand what this variation means for their ability to interact with plants and promote their growth. And so pretty much all of the traits that a bacterium has that live in the soil are related to their ability to promote plant growth. So there's many steps that they need to take to ultimately end up on the plant roots and carrying out a beneficial function. So they first must kind of find the plant root, then they must be able to establish there and grow, and then ultimately be able to carry out these beneficial functions. And so I guess I wanted to understand more about which traits are most important or most indicative of beneficial strains, and in particular which genes. And then once we have this knowledge, we will be able to predict which strains are most beneficial just by looking at their genomes. And so I'm really fortunate that some other researchers in Germany have made this tool that is like a database of all plant-associated traits from a range of bacteria, including Azospirillum. And so I use this tool to predict plant-associated genes in Azospirillum. So I've got a huge list of all of these genes, which is really exciting. And then so I wanted to understand how the strains vary and the number of type of genes for each trait. This analysis is very recent and still ongoing. It's got a big data set to work through. But essentially, when I plotted, again, just to not keep you in suspense, when I plotted genome size against the unique number of plant-associated genes that are predicted, you can see there's kind of roughly a correlation. So I guess strains with larger genomes have more of these plant-associated genes. which suggests that maybe having a larger genome is beneficial. But then the next step will be to determine which genes are encoded and then how many copies of each gene do they carry. And then the next step after identifying these traits at a genetic level is to understand the functions that they are predicting. And so this will involve carrying out phenotypic assays for these traits and then ultimately to link these phenotypic variations to the genetics. And so I've been using two different traits initially to look at this in more detail. The first trait, I won't go into the details here, but essentially the main mechanism through which azospirillum is predicted to promote plant growth is through the production of indole 3-acetic acid, which is a plant mimicking hormone. And essentially it stimulates plant root growth so that the plant has an increased surface area to absorb nutrients and water. And so there's multiple ways that the bacteria produce this. They are mostly dependent on tryptophan. It's part of the biosynthetic pathway. There's also tryptophan-independent pathways. And I worked with Chris Mitchell in the CDC metabolomics department to carry out a kind of simplified version of a HPLC experiment to look at the production of this particular plant hormone. And this is preliminary data that we generated a few months ago. You can see that they're all orange bars. It's the process that the IAA that we observed is dependent on the presence of tryptophan to be synthesized. And there's also variation between the strains. So suggesting that genetic differences may be leading to these phenotypic differences. I also worked with an undergrad student to look at another important plant-associated trait. So phosphate is a limiting factor in the soil for plants. It's kind of locked up in minerals, so plants can't access it unless there's certain bacteria around that produce organic acids to kind of liberate it and make it bioavailable. So I use this particular growth media here to study whether my bacteria strains are able to solubilize phosphate. And so these are just the pretty agar plates that we produced for this project. Each of these dots represents a different type of bacterial strain. And you can see that there's, again, variation in the phenotypes. But also the important thing to look for here is the growth of the colony. Some bacteria prove better than others. You can see with this one here, there's this clear zone surrounding the colony, which means that it secreted acids into the environment that were able to solubilize phosphate. So this is an example of a really good phosphate solubilizing bacteria. Again, fairly early stages, but there's a lot of exciting plans in the works for kind of determining which traits, which genes are important, and testing these in the lab, but also with plants. I guess the last thing that I want to talk about quickly is the work that I've been doing to establish a plant bacteria model system, and to do this by kind of looking at whether these bacteria are generalists for all types of cereal plants, or whether they can specialize to certain plants and maybe they're more beneficial for some plants than others. And so, as I mentioned, the composition of the root exudates determine which microbes assemble to form the rhizosphere microbiome, which means that some bacteria might grow better on some plants than others. And there's evidence on that, but there's also evidence that azospirillum associate with diverse plants, suggesting they're kind of generalists. But in addition to that, there's a small study showing that certain strains are more beneficial for certain plants over others. So it's kind of unclear whether they're specialists or generalists. And so I recently got funding from the Royal Society to carry out this large kind of pilot experiment where we grow. I also got a funding for a research intern, and Amy's joining me for the summer to work on this. And so what the plan is to grow 5 different types of cereal plants and perform plant growth experiments with the library of azospirillum strains to compare how well each strain can promote the growth of this range of cereal crops. And this will involve measuring plant growth by looking at the root and the shoot volume and weight. And part of this grant was to get a plant root scanner and software, which is an amazing tool that's been developed by a country in Canada. And essentially you can clean the plant roots and shoots. These are the pictures that I generated. That's not my scanner. And the software is able to tell you the area and length of these systems, which is a really useful kind of unbiased way to measure these plants. Yes, so to summarize, looking at soil health and in particular focusing on the soil microbiome will be an important focus for sustainable agriculture in the future, but there are a lot of unanswered questions. But I'll argue that studying model organisms can provide really useful insights into how these microbes function. And Azospirillum is a really useful model to learn more about this complex environment, and in particular looking at bacterial evolution and plant-bacteria interactions. And so thanks for your attention. I'd like to thank the Westra and Van Houten groups. It's amazing that I still get to work with you all, and also the ESI community. I really like working here, so thanks.
Okay, do we have any questions?
Bridget, you mentioned in your sample how you put your new Azospirillum strains that you collected them from the rhizosphere, but also from bulk soil. Have you tried to see whether there's any differences in the patterns between these two different habitat types?
Any difference in the what, sorry?
Between bulk soil and your rhizosphere Azospirillum, they're different traits. Because that was one of your predictions, that the very nutrient-rich rhizosphere would support or would have more defense systems.
Yeah, no, exactly. I kind of hypothesized that, you know, getting the rhizosphere sample, I'll kind of have an enrichment step, environmental enrichment step. I haven't looked into it exactly. I'm currently still isolating more from the bulk soil. So when I have my big data set, I'll definitely look at it, and also just kind of see if it's kind of random, the number of traits, or just like, you know, their defense systems. But definitely an interesting question about whether it was actually an enrichment step or just random.
Slightly less highbrow question. As kind of amateur gardeners, is there anything that we can do ourselves to improve the soil health of our own gardens? Basic things that we could do, practices.
Yeah, I mean, great question. Slightly unlike my own house, but I mean, from the perspective of a microbe, it wants, you know, lots of diverse plants to make, you know, diverse kind of soil environments. Lots of different microbes are kind of supported, and you know, making sure that kind of trying to minimise the overturn of soil, just, you know, be gentle when the weeds out, that sort of thing. And, you know, making sure it's rich of compost and lots of nutrients, that there's lots of substrate for your microbes. You can actually buy a lot of additives that you can, in the US, you can buy Azospirillum to add to your garden. But yeah, I guess just trying to, diversity is a key component.
Sorry, keep going.
Just coming at it because from a farming background, following on from that, how long does Azospirillum last in the soil once you've removed the plant?
Yeah, that's a good question. I think the results are a bit unclear. So some studies say that even when you establish in the rhizosphere, it would only kind of hang around for the initial phase of the plant growth. You know, it kind of does its job of increasing the plant surface area, and then it is outcompeted by other microbes. Long term, I guess it's again not clear, but they exist in the soil. So I guess that's why I was isolating strains from the UK. So maybe when we add them, they will be more persistent because they're adapted to those conditions. They're not one of the dominant taxa that exist in the soil, but you know, I guess they're there, and I'm sure when a plant comes along, will rise to the challenge and be useful.
So should we be looking at keeping them or increasing the numbers of them rather than adding them?
Yeah, I mean, that's it. You know, there's lots of these ideas about how to use these, and people apply lots of different approaches. But that's definitely,
Yeah.
how do we support the existing microbes? I'm not sure if there's a clear way how we can do that, but hopefully by looking at the host range, there might be a particular wheat cultivar that, better suited to Azospirillum, and a lot of this plant bacteria interaction. A lot of people are kind of approaching from the plant side to look at, can we breed plants that produce particular root exudates that then select for the most beneficial microbiome. So lots of unanswered questions, but lots of exciting areas and ideas.
I just had a crazy question. So when you were showing the soil with the plants and the bacteria kind of, accumulating around the plants, I kind of thought about the ocean and how you have like different little islands and a lot of the species accumulating around those. Is there anything that we know of, like, community assemblage around, and the phenotypes that are required of the fish or whatever crustaceans or whatever assembles, that we can use to think about key phenotypes that might be important for, one for those assemblage of bacteria?
Yeah, it is a great analogy, and lots of like microbiology is kind of known about the ocean because the soil's harder to study. I guess the environmental trait will just kind of select for whichever it needs. But if we can kind of manipulate it, and good analogy.
You have any questions online, but we're almost done. Thank you so much, Bridget. That was a great talk.
Thank you.
Thank you online for joining us.